Every process that is being carried out inside our body is well regulated. Now, that's why the brain is present here, and that is responsible for the integration and regulation of every function inside our body. So, like just like other functions, the formation or synthesis of insulin is also well regulated. Obviously, there is a time when there is a need of high insulin inside the body, and there is a time when there is the need of the low insulin inside the body. So, how it occurs? What is the thing that triggers the synthesis of more insulin, and what is the thing that decrease the synthesis of the insulin? As I told you that there are the main stimulus that is responsible for the synthesis of insulin is the glucose and the amino acids that are present inside the body. When their level is high, more insulin is released, and when their level is low, low amount of the insulin is being released. Now, that is the simple regulation of the insulin, you can say, or a simple balance between the insulin and the glucagon. Now, this balance is also regulated by some mechanism behind, like there can be the regulation of enzymes or activation or suppression of any structural modification or genetic modification that is responsible for the regulation of the insulin. Now here you can see as the insulin is a protein and it is being synthesized from the ribosome, and ribosomes involve transcription and the translation. So the main thing or the core process that can be regulated here is the transcription and the translation. So we can say that the factors that are responsible for the regulation of the insulin are the transcription. Transcription is basically the synthesis of, or you can say, the copying of the DNA to form the messenger RNA. So messenger RNA is produced in the transcription, and then messenger RNA stability, it also determines the amount of the insulin that is being produced. What is messenger RNA stability? Messenger RNA is produced and messenger RNA is degraded. Now these are two steps that take place in the body. Now, messenger RNA stability is basically the gap or the time duration in which the messenger RNA is produced and then decayed or degraded. So the gap between the production and the degradation of the messenger RNA is the messenger RNA stability. So if there is the long stability or high stability, then what will happen? There will be the most release of the insulin, and if there is low stability, less amount of the insulin will be synthesized, as again the translation process takes some time. Then there is the translation. Now translation is basically the transformation of the protein or the information from the messenger RNA to the formation of the protein or the amino acids. Now this translation can also be regulated to form to regulate the level of the insulin inside the body. And then there is the post-translational modification. Now post-translation modifications that takes place after the synthesis of the insulin, like the addition of the signaling molecule, then the conversion of preproinsulin into the proinsulin, etc. So these all post-translation changes or modification can also be regulated through enzymes and the genetic pathways.